Hello, everyone, and welcome. Thank you so much for attending our OTS webinar series. Today, we have two speakers. So we have Tracy Mallette and Jeremy Hall, who will speak. Tracy will speak first, and then Jeremy. And so we'll set it up so that we'll have our speaker, and then we'll have a question period, and then we'll transition into our second speaker. And at the end, we'll have some brief announcements of some upcoming webinars. So stick around. Also, if you have to leave at some point or you're interested in sharing the talk with your colleagues, it will become available. We're recording the session, and so the panelists, the speakers will be recorded, but none of the participants are recorded. And so if you have questions during the chat, do feel free to ask them using the Q&A function, and then we will moderate the questions at the end of the talk. All right, Tracy, you want to go ahead?
Yep, thank you. Hey, good morning. Thank you so much. I'm super excited to be here and present my work on Triblock heterochiral oligonucleotide nanotechnology. So first, let's talk about the molecular biology of life on Earth. All life on Earth shares several fundamental chiral traits. DNA and RNA are homochiral, containing only D enantiomers, and likewise, L amino acids that are used in biological production of genetically encoded proteins are only L amino acids. This chiral selection is really striking when you consider that abiotic production of chiral molecules usually produces a racemic mixture of the different chiralities, which leads to the question of how did life on Earth end up with this chiral dominance? Well, new research that has studied amino acids found on meteorites has discovered that there's a chiral imbalance already present with these building blocks, which suggests that chiral selection may have began before the beginning of life on Earth. What's the impact of this chiral dominance? Well, Newman's principle of symmetry can be restated as the absence of mirror symmetry of molecules has a remarkable effect on their physical and chemical properties and functions. So in gourd plants, what this might look like is the homochiral sugar unit builds into structures with the same handedness all the way up to their tendrils. And for me, what I'm most interested in is that the dechirality of DNA and RNA have led to proteins and enzymes that only recognize that chirality. So using nucleic acids of the opposite chirality should open a pathway to circumvent degradation caused by this molecule. So LDNA is the enantiomer of natural DNA. It's a left-handed double helix, and ddNA and LDNA have many identical properties. They hybridize the same way, there's the same duplex thermostability, the same cellular toxicity, but most importantly, LDNA has high nuclease resistance because of that chiral dominance that we just discussed. So with the laws of mirror symmetry, Watson-Crick base pairing occurs within each chirality, but if there's a chiral mismatch, like LDNA to ddNA, it doesn't bind predictably. So this creates a really big challenge when you want to use this left-handed molecule for performing any functions in the cell. And so our solution has been to create these heterochiral translators. These are DNA strands that contain both D and L chiralities, and it allows for the hybridization of matching chiral sections. And there's an image of what, an artist might think this could look like, where you have a strand that's turning in one direction until it hits a chiral interface, and then it starts to turn back in the opposite direction. And what do we want to do with these molecules? Well, the goal of my work is to create a robust bioorthogonal system for intracellular molecular computation that can sense endogenous inputs and produce outputs capable of interacting directly with these biological systems. So how do you do this computing? Well, there's many different strategies, but the one that I use is called Toehold-mediated strand displacement, or TMSD. So at the top of this figure, there's several different abstractions of DNA, starting first with the DNA double helix structure with some overhanging single-stranded unbound sections. And you can also look at this at a sequence level or you can reduce it even further to what's called a domain level. And this is kind of the representation that I usually use. And so when you look at TMSD, it first starts with toehold binding of these single-stranded portions between complementary sections. After this, the incoming strand begins to displace the bound incumbent strand through a process called branch migration. With any DNA duplex, single nucleotides will spontaneously dissociate from and rebind with their complement in a process known as breathing. Branch migration takes advantage of this breathing to move the junction between the bound domain and the incoming strand down the duplex until finally strand displacement is complete and you're resulting in the incoming strand being fully bound to the complement and an output strand being released out into the solution. So for what we usually talk about, this incoming strand is called an input, and the strand that's released into the solution is labeled as an output. So let's look at an example of using TMSD to implement the logic function AND, where two different input signals must both be present to release the output signal. So first, your input A is going to come and bind your gate. The gate is the duplex that's already been formed at the one domain toehold, and it's going to undergo toehold-mediated strand displacement, and then you'll end up with this byproduct duplex, and you'll have your gate now exposing the 2 and 3 domain toehold. Now your second input B can come in and bind at this 3-star toehold. The star designates that that's the complement to the domain, and undergo TMSD again until now you have this new output C, which has a different sequence of domains released into the solution. And this output can either then interact with another gate, or it could perform a different function. And so by layering different logic gates, and or not and, there's a lot of different functions that can be coupled together and combined in a cascading circuit for complex information processing. So you might have any different combinations of inputs that you can design to result in an on state, and if that combination isn't present, then it'll be in an off state. And this becomes really useful if you want to think about distinguishing between perhaps different sets of cellular markers. So going back to my overall goal, if you look at chemical modifications that have previously been used for nuclease stability, these often change the DNA hybridization rates, which can significantly affect the kinetics and functions of those strand displacement-based logic circuits that we just talked about. So really the advantage of my heterochiral translators is it could allow decades of DNA computing research to be straightforwardly implemented in degradation-resistant LDNA. So in order to accomplish this goal, we first had to build a chiral translation system. So we built these sequences on a leak-list design principle from Wang in 2018. And it uses 2 double long domains in order to start in one chirality and end in another. So what's happening with our circuit is we have an input which we've labeled X, and you can imagine that everything that's red is ddNA and everything that's blue is LDNA. So after this first TMSD with this gate, now we have an intermediate strand that's kind of half ddNA and half LDNA that then can interact with a second gate and release an output Y, which is now almost fully in LDNA. And this could interact with a FRET-based reporter so that when this Y molecule and the reporter molecule come together, now we have fluorescence, which can allow us to monitor this reaction. So we built this system, and this was the first time that toehold-mediated strand displacement had occurred across a chiral junction like this. There's been some early work in the 90s building, heterochiral chimeras. There's been a little bit of work before this using them in TMSD, but it had never really done anything where you go across this chiral interface. So it was a bit of an unknown of whether or not there would be some sort of kinetic block where this couldn't happen. But we designed this system and we tested 3 different formats of it. We have, I designated input to output chirality. So we go from the L domain and we translate into the D domain, or the D domain into the L domain. And we use a fully ddNA as a homochiral control. And so if we take that system with the 2 different translator gates and we trigger it with input, we get a fairly linear response. And we're able to show that we're able to go back and forth between these 2 different chiral domains. We did test this chiral system to make sure that it was specific by either mirroring the input to the system or swapping out one of the gates of that translator cascade with a homochiral control. And so through this, we were able to show that this circuit only proceeds as planned if you have every piece designed and input with the chiralities that we would want. But really what we wanted to see was how does this perform in the presence of nucleases? So we used serum, a mock biological fluid of 10% fetal bovine serum with DMEM in order to have active nucleases and everything that's needed to support them. And we started incubating this circuit at 37C. And you can see that if it's all ddNA, you get a really rapid amount of leak. And if you trigger the input, you still get response at 6 hours. But what's really notable here is that going from D to L chiral domain, there's very low leak, and then you still get a system response when you trigger in that input. And if you look at this across 24 hours, you can see that the D to L system, although it does have some leak, is significantly better than the homochiral control. So this was the establishment of the heterochiral translator circuit that my current work is built on. And it demonstrated signal translation. It was the longest cascade of chimeric gates to date, and the first one that went across that chiral interface. But what we really noted and what we wanted to do was improve the system response of the chiral translation system. So anything that's going to be sensing endogenous inputs needs to be very sensitive, because these usually occur at pretty low copy numbers in the cell. So after we demonstrated the successful chiral translation, we wanted to be able to improve that signal response. And what we thought was happening for this diminished response was, well, we're going from D to L, and so we're robust against nucleases in the downstream, but the upstream part of the circuit is not protected. And so when nucleases degrade it, now there's no longer anything that can recognize that input. And this led us to our hypothesis that we could use Triblock heterochiral oligonucleotides to solve this problem. So if you have exonucleases that can degrade ddNA but can't degrade LDNA, can we use this pattern of L and then D and then L again to protect against exonuclease mediated degradation, which should keep our upstream intact so that we have a low rate of leak and a high rate of activation. So these protective motifs, where we refer to them as toecaps, as they're protecting the toe holds. We know that most exonucleases degrade from the 3' terminus, so we wanted to design a couple systems to see, does every part of the circuit need to be Triblock, or can we use less LDNA and still protect our circuit? So we incubated our translator circuit with either 3', 5', or 3 and 5' toe holds in the same 10% FBS and 40% DMEM, and we monitored for 6 hours before triggering in our input. And so our blue line here is our control without any toe caps, and you can see that protecting the 3' toe hold really improves that system response. And what was most notable is where we look at our activation to leak ratio. So leak is unwanted positive background signal. And with our control system, we were able to get a bit over a twofold difference between the activation and link. But if we protected our 3' and 5' ends of our strand, then now we're getting close to 7-fold difference. So a really big improvement. We designed these LDNA Toecaps to be non-interfering with the LDNA portion of the circuit, but we thought that this should be independent of sequence. So we tested a 5-nucleotide poly-T Toecap as well as a 5-nucleotide poly-A Toecap and found that they responded exactly the same. So this was all protection of DNA duplexes, and we wanted to look at how could we use this Triblock heterochiral DNA as inputs. So we thought we could extend this, that, well, we've shown we can protect our circuit. Can we also protect that upstream input? A lot of different DNA circuits use single-stranded inputs as fuels or to trigger different parts of the cascade. So this was a pretty relevant test to do. And everything we did previously was with 5 nucleotides of LDNA. So that was effective at protecting the gate duplexes, but is that length really necessary for this single-stranded input? So we designed 4 different inputs. We had our homochiral control, which had no protection, and then 1, 3, or 5 nucleotides of LDNA. And we compared the ability of these inputs to trigger our translator system after 0, 4, 8, 12, or 24 hours of incubation at 37C in serum supplemented media. So all of the incubations happened first, and then it was added to our translator gates, and we monitored the reaction, and I'm showing the endpoint fluorescence of these systems. So what's really notable here, so our control is this teal line, and after 4 hours, there's very little left of this strand that is intact enough to trigger Toehold-mediated strand displacement. But if you add just 1 nucleotide of LDNA, you recover over half of the ability of that input to trigger the circuit. And this was kind of really surprising that so little LDNA could provide so much protection. And then as you continue out into the longer term, now you start to see an advantage of having more LDNA on these inputs. So all of this initial work was done in vitro with a mock biological fluid, but of course we wanted to move towards more biologically relevant. So we tested these Triblock heterochiral inputs in HeLa cells, and we used a 1-step TMSD circuit just to measure the intactness of the strand, not really to complete any sort of logic circuit. And so we started by transfecting in our input into our cells and incubated them for 18 hours before we transfected our ddNA reporter. And this was really to minimize the amount of time that that weaker ddNA reporter complex is in the cells. And then we measured the fluorescence using flow cytometry. So our negative control received an off-target input during the first transfection, and then the reporter second. And all of our inputs were transfected in at 5 times the reporter concentration. And what you can see really nicely here is that adding these Toecaps to our input provides a significant farther shift in our population into the positive. We repeated this experiment 3 different days and found that the fold change of the positive over the negative is significant, that the Toecap is better and more intact. We confirmed these results doing some microscopy imaging, and just from a top level, our negative, you can see just a little bit of fluorescence, which is all going to be coming from leak of that reporter. You see a little bit more activation shown in green with our unprotected input, and then far more with our toe-capped input activating these reporter circuits. And if you look closer, zoomed in on these cells, you can see that they're in different spots in the cytoplasm as well as in the nucleus. So With all of this, we've shown that the formation of triblock heterochiral DNA strands and duplexes provides improved stability against nucleases, that these LDNA toecaps can protect single-stranded DNA, and that a majority of the protection is conferred by adding the toecaps to the 3' terminus, but protection on both termini provides the best system response. This toecap protection is independent of sequence, and the toecap length can be reduced for short-term protection, even down to 1 nucleotide of LDNA. And finally, that these triblock heterochiral DNA inputs are also more stable in living cells than their homochiral control. I want to leave with just one idea for future heterochiral strand nanotechnology, which is we kind of focused on this in the first part of the circuit goal. But I think it'd be really fun to work on the end and design and test a heterochiral antisense oligonucleotide and see, does this protective LDNA change the ASO knockdown efficiency? And also really going with that goal of linking in the ASO to the output of a molecular circuit computation. I'd like to thank my co-advisor, or my co-author and my advisor, Matt Leakin, the co-authors that worked with me on this work, NSF and NIH for funding, and also TS for having me talk, and I'm happy to answer any questions.
Great, Tracy, that was an excellent talk. I want to remind our participants that you can put questions in the Q&A, but I have a couple of questions just to start off. So this is, I guess our interests or expertise overlap in DNA nanotechnology. And so my questions are, this kind of strand displacement strategy is incredibly effective and has a lot of potential for all of the applications you've named, as well as biosensing and different systems, DNA computing. We can expand with the idea of, you know, instead of 1s and 0s, we have 4 bases. So all of these different sorts of applications exist. And so I'm wondering if you kind of, in your experience in this research, what your strategy would be, or what advice, kind of looking back on the experience that you have for designing these sorts of hybridization systems, what advice would you have for someone who's interested in getting into this sort of research?
There's a lot of tools available for designing these systems. NewPack is great and it's available online. There's some different available tools like that and a lot of already existing circuits. And the nice thing about using this strand technology is you don't have to reinvent the wheel every time. So there's a lot of literature out there, and that's kind of, you know, our goal of using this LDNA for it is that if you're going to start introducing like phosphorothioate backbones, those can really significantly change the way your circuit's going to go. So making sure that you understand those kinetics before you introduce it in, as well as, you know, trying to come up with clever ways to not use so much of those modifications so that you can have a more straightforward approach. But I guess my advice would be look at what's already out there, because it's a really kind of deep field of what's already been designed.
Sure, that's a great answer. I'm also wondering too about, I mean, you showed this in your results, that you looked at kind of different lengths of these toecaps and of the strands themselves. And so did you find kind of any general rules or kind of limitations in designing these systems that you could kind of share with others to kind of help give people a head start?
Yeah, so I think the different lengths really is going to come down to what your goal and target is. So the nice thing about being able to design shorter ones is you don't always want everything to remain active indefinitely in these sort of cellular systems. So if your target is shorter, you know, you only want to be active for a little bit of time, you know, short amount of protection. But really the longer protections start to provide benefits as you go even deeper, and looking at how these protections will interact with your circuit, as well as making sure that your considering that you're only protecting against exonucleases in this case. So any endonucleases can still recognize your target. That's still why these have some sort of drop off, and screening that section really well, or evaluating what sort of additional protections you might want to add in there.
Great. That's awesome. Does anyone have any questions before we move on to our next speaker then? All right, if not, I just want to thank you again, Tracy. That was a really cool talk. Brought something kind of new and different, which is always inspiring. And so we're thankful for that. And so I'll pass things over then to you, Jeremy.
Okay, let me just. Okay, can everyone hear me and see everything?
Yeah, looks good.
All right, awesome. All right, hi everyone. Thank you for being here today. I'm Jeremy Hall. I'm a graduate student in the Toehold-mediated laboratory at City of Hope in Los Angeles, and I'm excited to talk to you about our work in developing a new application of oligonucleotides to cancer therapy, and providing some updates to the work that I last presented at OTS. I know we obviously started with some chemistry here. Actually, we'll be moving more into biology and stuff, but I will obviously go over plenty related to oligonucleotides. So just first things first, of course, no financial conflicts of interest to disclose. So the transcription factor STAT3 is one of the primary focuses in our lab. And as seen here in this diagram, you have the canonical pathway in which STAT3 is activated via phosphorylation by Janus kinases, which in turn are activated by cytokines and other hormones such as IL-6 and IL-10. And then once activated, STAT3 then forms homo or heterodimers, which can travel back and forth from the cytosol to the nucleus, and obviously they initiate a wide variety of downstream gene expression. So STAT3 has been considered a pretty attractive cancer target because of its known roles in tumor progression and immunosuppression in tumors and tumor-associated immune cells. In those cells, it tends to show aberrant signaling, constitutive activation that leads to increased expression, the likes of VEGF, IL-10, FOXP3, PD-L1, and a lot more. So in turn, obviously the increased expression of those cytokines like IL-10 and IL-6 then ends up leading to like this vicious cycle where STAT3 remains activated, hence that constitutive activation. But it's also particularly challenging to target STAT3 because there is a lack of enzymatic activity that we see with other traditional, more druggable targets, so to speak. And then there's also the fact that STAT3 is still needed for healthy cells. Notably T cells, it's a critical component of their growth and maturation. Suppressing STAT3, in targets like that would obviously lead to, severe immunosuppressive effects that we don't want to see. Obviously, universal inhibition of STAT3 is just simply not an option here. So our lab has been focusing on applying oligonucleotides to target STAT3. And in this case, we're trying to catch STAT3 in the act, so to speak, in the cytosol. And we did this with synthetic DNA as a STAT3 decoy, has a hairpin design to enhance the serum stability. A CpG motif is then actually applied to the 5' end to promote delivery specifically to TLR9 expressing cells. Those obviously would include B cells, dendritic cells, obviously various antigen presenting cells, and a variety of cancer cells, particularly those related to lymphoma and leukemia. And so previously published data from our lab showed that this oligodeoxynucleotide can be rapidly taken up by myeloid cells. It can interact with STAT3 in the cytoplasm, interfere with its nuclear translocation, inhibit STAT3 activity, and ultimately show a dose-dependent reduction of STAT3. And in addition, they were able to show that the CpG motif caused some immunostimulation that brought interest from an immunotherapeutic standpoint. But at the end of the day, those decoys actually have short-term, temporary, reversible activity. So in an effort to essentially take this up a notch and bring about irreversible degradation, we decided to apply the decoy oligo to a PROTAC model. So if you're not already familiar with the term, PROTAC is short for proteolysis-targeting chimera, and it utilizes a unique heterobifunctional strategy where there are two ligands, one that binds to the protein of interest, and another that recruits an E3 ligase. The common ones include cereblon, the von Hippel, VHL are pretty common ones. And then these two ligands are then held together by a chemical linker. And then ultimately the E3 ligase complex and then the protein of interest are brought into close proximity. That protein can then be polyubiquitinated and then set up for proteasomal degradation. So in our design, we settled on applying thalidomide to the 3' end of our STAT3 decoy, and then we set about optimizing the linker length. And the results from that seem to show that it's kind of a sort of Goldilocks situation, where if you have a linker that's a little too short or a little too long, it probably doesn't allow for the right amount of space and the right conformation to effectively ubiquitinate your protein. But in this case, the single linker unit here hits the sweet spot. Ultimately, we have the greatest reduction of STAT3 so far. So we then set about developing some in vitro models to demonstrate the actual mechanism of the Oligo-PROTAC. So we first used this non-CpG PROTAC that can be easily be transfected into mouse dendritic cells. And then we gained early promising results that showed a dose-dependent effect of the decoy. And ultimately, the PROTAC here shows a greater reduction of STAT3 compared to an equal molar amount of the decoy. And then since STAT3 is also known to have a lot of homology with various members of its family, particularly STAT1 and STAT5, we wanted to test for them to see if they were going to be affected by either this decoy or obviously the PROTAC itself. And we found that they were largely unaffected, indicating that this is very specific. It has high affinity for STAT3. That's obviously important too, because with STAT3 versus STAT1, STAT5, there are various downstream implications. STAT1, targets are going to look a little bit different from STAT3, and obviously STAT3 is what we really want to focus on here. And so while it's not actually pictured on the slide here, we also conducted some competition tests. Ultimately, they would include increasing amounts of either ligand, either the decoy or thalidomide. And ultimately, we were able to show that it was not some non-specific effect. It was actually the mechanism from both of these particular ligands that was actually reducing STAT3 levels. And then furthermore, probably one of the more important parts in showing this mechanism, we were able to show that concurrent treatment with the proteasome inhibitor, MG132, essentially eliminated the ability of the PROTAC to reduce STAT3 levels. So obviously this indicated that this was some proteasome dependent level and indicative of proteolytic degradation. So moving on, we began testing the full design that includes the actual CpG motif. And then at this point, we were able to do free uptake in vitro models, the same kind of cell lines. So in this, we have equal molar comparisons ultimately show that the PROTAC continues to bring about the greater STAT3 reduction than an equal molar amount of the decoy. And it still demonstrates that dose-dependent effect that we saw in the previous slide. And notably, it does seem to hit about 50% inhibition around the concentration of about 250 nanomolar. And also not shown on the slide here, but we did also conduct this in other cell lines as well. We did it with both mouse and human lymphoma cell lines, notably H20 and OCI Li18, and we continued getting some similar results. So testing in primary mouse cells actually showed that STAT3 was reduced in B cells and myeloid cells, but not in T cells. So it was obvious that the CpG motif promotes uptake in those specific cells that express TLR9 and certain scavenger receptors. Just obviously note that the STAT3 levels here in B cells and myeloid cells, it's indicated by the top band. Obviously, that'd be significantly different from that for the T cells. And then we were able to demonstrate that there is also some resulting immunomodulatory activity when targeting STAT3 with these dendritic cells, and ultimately showed like a significant reduction of these pro-inflammatory cytokines that are associated with myeloid-derived suppressor cells and immunosuppressive tumor microenvironments. Ones that are like IL-6, IL-10, IL-1, IL-12, GM-CSF that are known to recruit and ultimately result in these immunosuppressive environments. So in this model that was generated in solution for us by our colleagues in the Department of Computational Medicine at City of Hope, can actually see the decoy ligand interacting with the DNA binding domain here of STAT3, which in turn, obviously, and then obviously we have thalidomide on one end. Obviously, it's a little small in this diagram. Obviously, as a small molecule, it's a little bit hard to see here. But in turn, it is recruiting cereblon, which then in turn recruits the remainder of this E3 ligase complex. So to more specifically illuminate this mechanism, confirm it, we again used dendritic cells and knocked out cereblon, and essentially found that knocking out cereblon, more or less abrogated any effect of the PROTAC. Obviously, it appears to confirm confirm that thalidomide is attracting cereblon, and that it's obviously a critical component of bringing about the remainder of the ternary complex here. And so our colleagues also presented us with two lysine residues that are seen up here, lysines 601 and 626 as likely sites for ubiquitination based on the ternary complex that's caused by the PROTAC. And ultimately, the final relative location of RBX1, which would be what actually transfers ubiquitin to those STAT3 lysine residues. And so we found that mutating those residues to alanine actually eliminated the efficacy of the PROTAC. Mutating just one at a time at least reduced it, if not, totally eliminating its efficacy. So it at least appears, at least with this, the specific PROTAC, that both these particular lysine residues are a critical component in the degradative mechanism here. Whoops. So we finally began moving into in vivo. So this is our newest data. We finally began moving into in vivo models, developing one here that involves NSG mice that were engrafted with subcutaneous OCI-Ly3 tumors. And then these mice then received intratumoral treatments where we would compare the PROTAC against the original unconjugated decoy, as well as a scrambled decoy that still has the CpG motif. And our results ultimately show that, obviously the original decoy could still slow the growth of the tumor itself, which was consistent with what previous experiments had shown, and that, the CpG scrambled was also still demonstrating a very, weak, but still a slight effect of, ultimately tumor growth overall, which was possibly, attributed to its, TLR9 agonist activity. But ultimately, it's the PROTAC that demonstrates the most efficient prevention of tumor growth, and it's the only one that actually even shows any eventual beginning of a reduction of tumor burden here. And ultimately, our measurements of tumor mass and volume actually revealed that the PROTAC appears to have an anti-tumor effect that's about 2 to 3 times stronger than what it was for the decoy, and about 4 to 5 times stronger than it is for obviously the scramble and for just PBS alone. So obviously these are still early results, and we want to continue to confirm them in more models. We obviously want to really look into syngeneic models using like A20 cells in in bulb C mice. Potentially look more into what's going on in the tumor microenvironments and how that's changing and all that. But we are obviously excited to get these results pretty early on. So in conclusion, I think we've been able to, hopefully I've been able to show you that we can actually build on the original STAT3 decoy with this new PROTAC strategy. I think we've been able to show that we can upgrade temporary inhibition to more powerful degradation while maintaining the cell-specific delivery and the affinity for STAT3 that are going to be crucial components for actually delivering an effective therapy in the future. And we're able to see, I believe, improved anti-tumor effects compared to the decoy in an in vivo model, obviously with human B lymphoma cells, and what's known to actually be a pretty aggressive, highly mutated form of B cell lymphoma. So we believe this could, have very, legitimate potential in the future of treating B cell lymphoma, possibly acute myeloid leukemia, And perhaps most exciting is we believe this is a pretty flexible platform. We believe it can be applied to other targets that have traditionally been considered undruggable, looking at potentially other oncogenic transcription factors in particular. We're actually currently working on targeting NF-kappa B, and again, in a kind of similar activated B cell lymphoma model. And that would obviously be, particularly exciting if we could harness that PROteolysis technology to target something like NF-kappa B, which is really like the holy grail of immunomodulation here. So with that, I would like to extend a sincere thank you to everyone on this list, to OTS, the organizers of this webinar, for inviting me to be able to speak with you today. Obviously, all the people have an enormous wealth of resources here at City of Hope who have all contributed, an immense massive of knowledge and contributed lots of various contributions to this project. Most especially, of course, I'd like to thank my PI, Dr. Korleski, and everyone in the lab as well, who has provided insight and guidance along the way. Everything from helping design in vivo models to figuring out basic reagents for every basic kind of test. So with that, if we have any questions from anyone, I'd be happy to answer.
Thanks a lot, Jeremy, for this excellent presentation. As Jeremy mentioned, if you have a question for Jeremy, please use the Q&A function. But I can always start with a few questions. So I think in one of your first slides, you mentioned that there are quite some downstream targets of STAT3. So did you check the effect of your treatment on these downstream targets?
So the PROteolysis specifically, not in this particular paper, but we have with the decoy, we do know that MYC obviously is a significant, well, obviously, realistically, the Luminex data ultimately showed, a lot of these are definitely downstream of STAT3 as well. Like I know IL-6, like obviously but basically with STAT3, obviously we're talking about like a lot of, a very circular type of thing here. Like IL-10, IL-6 are definitely downstream of, they're both upstream and they're both downstream of STAT3. And then I believe we also have indications that MYC was reduced as well. Obviously that would be a critical component, obviously, in the cellular growth, at least obviously for these particular kinds of aggressive cancers. And like I said, GM-CSF, obviously all these pro-inflammatory cytokines and whatnot, which are known. And obviously that's, SOX3 is known to be obviously downstream of STAT3. So obviously it's, believed to be attributable to that. Like if you are targeting STAT3, you're obviously having some effect on SOX3, which in turn is having its effect on these cytokines as well.
Okay, cool. We have a question in the chat from Feng Guo. At what concentration your conjugate started to show cellular toxicity?
So in our models, usually, if it was transfected, it was usually showing somewhere between 10 and 50 nanomolars. And actually, I can go back in free uptake. We are seeing it in a free uptake. We were seeing it as with the PROteolysis, we were seeing it as low as 50 nanomolar. Obviously the largest amount we were seeing at least half of it. We were seeing at least 50% degradation around 250 nanomolar. But yeah, we can see it as low as around as low as 50 nanomolar or so. And in the future, obviously, like we never know. Like there may even with the evolution of like boron clusters for oligonucleotides and whatnot, we may even see lower concentrations that we're able to see with this.
Question from Jean-Jacques. Is there any toxicity associated with the thalidomide?
That we have seen so far, it's so obviously in this case, obviously when we did our competitive testing with it, I mean, there's realistically when we're testing a solution like this, it doesn't appear that it's, I mean, there's going to be some level of thalidomide, but we're not talking about free thalidomide here. We're talking about it being specifically conjugated. So like obviously the whole point of the mechanism is that it's thalidomide is actually like recruiting the E3 ligase, and then together it's working with the decoy. So it's not working, on its own in that way. Like when we did competitive results, when we had an increasing concentration of thalidomide against the PROTAC, it did eliminate, it gradually eliminated the effect of the PROTAC. So it does, it does appear to be dependent on both ligands, not just on thalidomide.
Okay, the question from Ming Ming. Great talk. May I ask, did you take the imaging of STAT degradation like with other techniques? I think you showed a lot of Western blots. Did you also use like immunofluorescence or whatever?
No, so far we're primarily looking, obviously, like at least in this model, no, like obviously with the previous decoy model, they had looked obviously into some confocal microscopy. That's how they were able to show that it was essentially interfering with the nuclear translocation, and that it was interacting with STAT3 at the cytosol. I can go back to that previous slide where they showed that here. Obviously in the top right hand corner, kind of see obviously the the decoy in that case was in red and interacting with STAT3 and blocking it from the nucleus, which was STAT3 obviously being shown in green there. But for this, we were primarily focusing on the actual, we used Western blot, to essentially get an idea of like the actual protein presence here. We're not necessarily targeting STAT3 RNA or anything here. We're actually targeting the activated STAT3. That's the point here. So we found the Western blot to be the most effective mechanism to be able to get an idea of like what concentrations were actually affecting activated STAT3. We found the Western blot was the most effective for that.
Yes. Then so just so I think in this particular paper you used IV systemic delivery has been used, and I think in the other slide that you showed later you used IT delivery. So is there anything known about biodistribution?
For the PROTAC, not yet. I mean, obviously the decoy itself was pretty well established. Obviously with the PROTAC here, we don't necessarily have all that information yet. This is still relatively early stages. We've just begun some in vivo work here. Obviously we'll be looking more into the distribution of how that takes place and all that. But yeah, obviously, like we'll potentially look into to IV treatments as well. For now, we're just starting with IT.
Yeah. Clear.
Obviously,
Yeah.
with subcutaneous tumors, sometimes it can be difficult as well too. So you want to make sure. In this case, we'll obviously want to make sure we can actually target those. And so IT treatment was the most reasonable beginning for that.
Yeah, and then I think the other question that's always important is it's always about how functional something is. So how good does it target your target, but also safety. So any ideas about the safety profile? I think what you mentioned is that you mentioned the other statins, so no effect on the other statins, but probably it might be good to do like a more rigorous safety.
Oh yes, obviously obviously in the future there will be more assessment obviously of the safety profile. There was a lot more about that with the actual original decoy. It was found to be pretty sick. The main problem with the decoy ultimately was that it was its activity was short term. So we obviously something we would need to look at into the future is was the decoy safe because its activity was short term, and if the PROTAC, if it's exerting a longer term activity and ultimately degradation, is what's that going to look like from a safety standpoint? So obviously, yes, they'll be looking to that in the future.
Yeah, and then I also had a question about cell type specificity, right? So I think there was I think your CpG motive clearly showed the difference in uptake in B cells versus T cells.
Yes.
So would there be, if you change this particular motive, would it be helpful for other treatment strategies? So just having another motive directing it to other kind of cells, would it be feasible with this particular strategy?
Potentially. Obviously, the advantage with TLR9, obviously in this case, is that it is pretty specific to what we're interested in here. Obviously, we're interested in B cells and targeting myeloid cells and avoiding T cells. And TLR9 was very advantageous in that, TLR9 is not that widely, expressed or anything like that. And the scavenger receptors that are also sometimes picking up the CpG motif is not necessarily present on other cells as well. Obviously, we would have to look into, like, when I talked about the flexible platform, we potentially would be looking into, other types of motifs, to deliver to other types of cells, other, indications and whatnot. Like, obviously, in this case, we're still focusing on lymphoma. And so it obviously just made sense with B cells and antigen presenting cells. But yeah, in the future, obviously, we could look into what it might like for, for anything about TLR5 and TLR7 and whatnot, but they are not necessarily as specific as TLR9 as well. So that is something that we'd have to keep in mind.
That makes sense, I guess. So another question from Cook. Thanks for the great talk. Have you tried to look at other proteins interacting with CpG STAT3 PROteC by proteomic in order to check inhibitors?
So like when we're treating with this, if there are other proteins that are affected, essentially. Yeah, so I believe NF-kappa B was a little bit affected. Obviously, there's there's going to be some cross activity between the two, and with these ABC, these activated B cell type lymphomas, you're going to probably see NF-kappa. That's why we're in the future also looking at NF-kappa B, because more likely NF-kappa B is going to be further upstream, could have more immunomodulatory capabilities. Obviously, the important thing that we really wanted to consider was this, because of the decoy's design, it's very specific. Like, it's basically binding to the DNA binding domain of the protein, and if there's just like one or two bases that are off, it's not going to bind, it's not going to bind to the transcription factor. It has to be very precise. These are pretty precise consensus binding sequences. I mean, if it's the main thing that we wanted to show was that there are proteins that are pretty close in homology to STAT3. That's STAT1 and STAT5, and it didn't really appear to be affecting with them. It is very specifically targeting STAT3.
Good. Well, thanks a lot, Jeremy. So I would like to thank both Jeremy and Tracy. And when I looked at the very last slide of Tracy, I think she's interested in a postdoc position. I think mainly for a combination between oligonucleotide therapeutics and nanotechnology. So if you have an open position, please think of Tracy, because I think she just had an excellent presentation today. So I would like to thank everyone for listening today, for participating in the webinar, and then the next webinar will be in 2 weeks. And in 2 weeks we have, I think, one of the greatest names in the OTS field. It's Mano. He's a Lifetime Achievement Award winner. So I think another great webinar in 2 weeks. So stay tuned, and I'll see you later. Bye-bye.
Thanks, everyone. Bye.